Well, good morning, everyone. I think we're ready to get started with our first session. Our first speaker this morning will be Professor Paul Yeager from the University of Washington, who is a professor and vice chair of the Department of Bioengineering there. He's going to talk with us about a point-of-care diagnostic system for the developing world. He began his career at the Naval Research Lab, where he was on staff from 1982 to 1987. Then he joined the Department of Bioengineering. Since 1992, his research has focused on the development of microfluidic devices for the manipulation of biological fluids. He was recently awarded a $15 million grant from the Bill and Melinda Gates Foundation under the support of the Grand Challenges in Global Health program, and I think he's going to talk about some of the work that's been done under that program today. So welcome.
Let me thank you very much for the invitation. I really appreciate it. It's fun. It's actually my first visit down to Rice. And since it's my first visit down to Rice, I figured I'd tell you a little bit about the technical issues. But before that, give you a little bit of context and how our Department of Bioengineering may be similar to your Department of Bioengineering, just to give you a little bit of context. So let's see if this works. This is an interesting Mac to Mac transfer here. We'll see if it works. First of all, we're a bioengineering department. We're older than your department, going back to '67, but in certain ways we're about contemporaries. Our undergraduate program is about the same age, and we're doing a lot of things trying to catch up, in fact, to what you've done as a wonderful job here in bioengineering, particularly with the undergraduate program. So we've done a lot of, we're a reasonably large department as these things go. But we used to be scattered all over the University of Washington campus, so we were sort of spread everywhere up there, and that was sort of fun. I was sort of up in this corner. This is the School of Medicine down here, this section down here of this enormous hunk of medicine research, and it's a wonderful place to be. We've just recently got moved about 12 months ago down there, which has put us in this lovely building, which goes by an interesting name, which is the William H. Fagey Building. And why is that important? Because you may have seen his picture as one of those famous people in global health. And I will confess that 5 years ago, I had no idea who this was, and we said, why are we going to have a building named by somebody we didn't know anything about? And then we have become educated. So my growing into global health has been a recent vintage, so please, no expertise in that area on my part, really. However, we're doing a lot of it, and actually had been before the building was dedicated, which was comforting. The building's, got a lot of money, $12 million from the federal government, $10 million from the Whitaker Foundation, and the biggest chunk, which includes our half as well as the Genome Sciences half of the building on the other side, came from the Bill and Melinda Gates Foundation, which, shall we say, really heightened our awareness of global health, because we had not realized at the time, but there was a coupling with global health in part of the building. So when they did the dedication, not only did we have Bill Gates to worry about, but we also actually got Jimmy Carter and Bill Fagey there. Bill Fagey is largely responsible for the eradication of smallpox, which was a very impressive story, and was head of the CDC under Jimmy Carter, and they're really good friends. And we had lots of statements about the importance of global health research in the Department of Bioengineering. So the pressure got torqued a little bit tighter on us, which was kind of interesting. So the question is, what am I talking about? What am I going to explain to you? I'll bet all of us today have these things in our pockets, okay? These things are really remarkable tools. We use them typically to make phone calls to find out, who's shopping for which thing at which supermarket to make sure we get the right stuff. But these are extraordinary communications tools, and they're an example of refinement of computer technology and data communications technology over decades. And to someone who started out thinking of computers as something you took punch cards to, this is a real revolution here in your pocket. And younger folks won't recognize this, but we certainly do. What is it? Well, it's a computer. It's a computer that can have downloadable software which allows you to put whatever program you want into it. It has a graphic display. Mine happens, and most of yours probably have cameras, which means not only can I look at digital images, but I can collect digital images and send them to any point on the globe. That's a huge amount of information that can be sent around rather easily. It also happens to have mechanical actuators. Mechanical actuators are tools you might use. And what I'm suggesting is, mechanical inputs, a whole bunch of stuff that I could use as a research chemistry laboratory. All it's missing, of course, is the interaction with the chemistry parts, and we'll talk about that. So what I've been worrying about for about the last 15 years is how to take small, portable technologies like this and couple them into the chemistries. So the chemistry part was sort of the weak link, and that's where the microfluidics comes in. So you can now look at the screen again. Bioengineers have been really good at working in centralized laboratories. That's our medical school over there, and it's a great place to go. It's sort of like the cathedral of the 20th century. It's the big edifice we build when everybody comes to it to get medical care. And it's really good for a variety of things like, results while you're there, but it's not much use for outpatient clinical things because you have to go there to get things done. We would like to suggest that what bioengineering is going to be doing in the 21st century is moving biomedical diagnostic technologies to the point of care, wherever that is, any place. Make them usable by anybody, not just a specialized technician, using small volumes of samples and reagents, low-cost disposables being the primary mode of action for these things. Getting a result from the time a sample goes in it in under 10 minutes for a lot of good reasons, which we think would enable a wide range of technologies and wide range of new types of research and treatment to go on in the developed world. And we think of this as, we call it distributed diagnosis in home health care, and I won't go through the cartoon in great detail. This cartoon's about 12 years old now. What's amusing is, of course, now we replace the laptop computer with the cell phone as the paradigm of the object that you can carry with you. And what I've spent most of my time working on is small plastic widgets that would interface with those things and do the chemistry. Lots of potential users, from hospital inpatients, clinical trials of new medications, monitoring hospital outpatients, which is the next frontier, looking at people with chronic conditions so they can take an instrument home with them to monitor the chronic condition, looking at critical drug treatment, monitoring pregnancies, early warning for emerging or emergent conditions, things like heart attack, stroke. So the patient would have with them something would say to them, I do need to go to the emergency room, or this really is just take a couple of aspirin and see me in the morning situation. And of course, anticipating, preventing, and managing outbreaks of infectious disease. So epidemiology as well as therapy and things of that sort. And that turned out to be, that last item was sort of I won't say the killer app, as it were, for microfluidics, which we've been working on. And of course, we're now thinking very much globally. And the real change in the mindset, which I think many of us agree is manageable, is that technology can also be applied to health problems in the developing world. Why would you say that when there are places with such low resources? How could you imagine taking the kinds of technology that bioengineers develop and moving to the developing world? Well, I am told by my friends who come back from Africa all the time that they can now get a cell phone signal almost any place they go, including places far away from big cities. How is that possible? It's a technology that's catching on because it does a useful thing out there. And what you often find in low-resource settings is that new technologies take over that you would never imagine get there because they didn't go through that stage of having the expensive big hospital infrastructure. And the fact of the matter is you can leapfrog technologies in places where the old ones don't exist that is even harder to do here, where you have to work through the existing and somewhat rigid architecture of the existing centralized infrastructure. So it's a real, in a sense, opportunity to take technologies beyond where they are now. Certainly, we all believe, SARS being a very good example that our Asian neighbors suffer through, that a health problem one place can rapidly become a worldwide one. However, whatever these solutions are for healthcare in the developing world have to be inexpensive. We've tried in my lab to focus on things that start out from inexpensive first and work out. Yes, we're techies, so we always think, gee, that'd be cool, I could do that. But what I'm trying to do as much as possible is say, wait a minute, given the constraints of a couple of bucks for this test at most, absolutely at most, how could we possibly get ourselves back to a technology that'll work? So we've been working on microfluidics for a while now, 15 years or so, which is basically working with pipes that have one dimension that's smaller than a millimeter. Very, very simple concept, nothing odd about it. They have the potential to automate complex processes. They're compatible with small sample volumes, which you and I would prefer to give rather than a vacutainer full of blood if we want to make a measurement at point of care. You can pack lots of tests in small spaces. There's little waste. You can integrate many components. In theory, can get reproducibility of function. And that turned out to be a critical one for the global health problems, as I'll explain in a minute. Potential for mass fabrication, potentially low inherent cost. You can mass fabricate something very expensive. But in theory, if you want to make something inexpensive, you'll have to use some interesting mass fabrication methods. So we've developed a variety of things at the UW over, again, this 15-year period, including things that separate fluids. And my website has lots of details, and I won't have time to go through them today. But let's just say that there are a variety of things, including devices unique to microfluidics, as well as an inexpensive lamination technique, which turned out to be a very good rapid prototyping technique, which basically meant cutting things out with a CO2 laser and stacking layers one on top of the other, that allowed us to go from an idea to a finished device in the lab in about 4 hours, which was, I don't know if it's the record, but it's a pretty short turnaround time for making devices. And that allowed us to make lots of mistakes. And believe me, we made them. We've worked with companies like Micronics, which is a spin-off company that came out of our lab to make more sophisticated looking devices. And I have a deck of these things here, which I'll be happy to share with you afterwards during the break. And we've also developed assays that are unique to the microfluidic platforms. Let me break out for a second, if I can manage to get the right button here. And I wanted to show you briefly a piece of a movie. This is something that Micronics did, but it's sort of typical of what you can do with the microfluidics alone with no instrument. And this was a card that was designed at the request of the New York Blood Bank people to, let's see, it's going ahead. This is designed to do blood typing, basically a simple ABO blood type. And you put a blood sample in. This one has wet reagents. The current version actually has blister packs on the back that are preloaded. So you wet those things out, and you use an onboard pump, which is literally putting your fingers down in two places, squeezing and releasing one thumb, which sucks both the reagents and the blood down across the card. And within literally 3 to 4 seconds, you've got an immediate identification of blood type. Okay, the whole card is aimed to cost maybe 50 cents in the US. Could be cheaper. Just a simple example of how relatively simple plastic microfabricated microfluidics can get you a useful medical result very rapidly. This is what the guts look like for those of us techie people who want to see it without the paper cover on. It's literally just filling by capillary action, and all the motivation for the fluids is just vacuum. You pull 3 fluids into 3 separate channels, and when they're sucked in there, they mix. Here's squeezing the bellows, closing off the port, and then releasing the thumb, which sucks the fluids down side by side. And it turns out these fluids mix very readily, and you get aggregation. Really simple idea. This is what I'll call the paradigm of the really simple device. If we could, we'd make all of the devices that simple. As it turns out, for some of these things, you do need some form of a reader. So we do a lot of our work on reader design and working with those sorts of things. But this is a very good example of a simple one. It's at what I'll call the near product stage. It isn't actually out the door yet. So what we really came to talk about is this Bill and Melinda Gates Foundation project. A wag at one of our collaborating institutions came up with the idea of DXbox for a working title, and it stuck because we liked it so much. I don't know, maybe you have Xboxes at home from Microsoft folks. We thought DX is, abbreviation for diagnosis or diagnostics, was a perfectly good idea of what we're trying to do. It was a device which would function to do diagnoses based on chemicals that you could get from a fluid sample. This is part of Grand Challenge 14, which is part of the Grand Challenges in Global Health initiative they put out. 14 was technologies that assess individuals from multiple conditions or pathogens at the point of care. It was one of those things where when the RFA came out, we looked at this and we said, that's what we do. And it was sort of one of these things that was written for us. It was actually fairly straightforward to plan out what we wanted to do. The tricky part, of course, was putting together the right team, which, as it finally ended up, has university people, myself and Pat Staten, nonprofit and corporate partners. So what is this thing we've designed? Well, this is a mock-up. Okay, this is sort of the classic, here's the wooden box that looks like what the final device is, with an instrument about the size of a bread box. People don't make bread boxes. A shoe box, a small shoe box is a good idea. It weighs about 9 pounds, including the batteries. Notice no cord. It will not need a power cord to operate. So we talked yesterday about what would be required to go out into the field to actually make useful measurements. It has disposables, which will contain all the chemistry, and all the water and all the liquids will be on the disposables. Put a sample in the card and run it. Okay? Take it out. The instrument's dry. All the waste will be on the card. So right now, the aim is to detect pathogens by at least 2 methods. Right now, we're designing it to work for PCR for amplification for nucleic acids, as well as immunoassays for detection of antigens and or antibodies. We're aiming at a turnaround time of 15 minutes per sample. okay, which means all the PCR and all the answers, all the things out, dumping a picture on the screen within 15 minutes per sample. So the instrument will be locked up for 15 minutes each time. Disposables has all the reagents, on-card calibration, and test validation, so you don't need to take it to the lab to calibrate the instrument. Yes, small, lightweight, rugged, battery-powered, something you can throw in the back of your Land Rover and take to wherever you go at that last mile, the periphery of the healthcare systems in the developing world, to be used by people of limited but non-zero training for the instrument. Simple operation as possible, and obviously low cost. And when I say low cost, well, where do we start for the instruments that go in there? We start here. Consumer products for which there are existing, very sophisticated pieces of technology that you can buy at the board level and use them and plug them into the box. As much as possible, we're stealing from the cell phone industry for the stuff that goes inside the box. End users, well, certainly there are many possible ones. We tend to think of disaster and crisis healthcare workers, small urban and clinics, and private and public caregivers throughout the developing world as potential users. One of the reasons the corporate folks are interested in this, of course, is there's another class of users, which are people in the developed world. And that's a separate and related problem. And if we want to talk about policy and economics at some point over lunch or a beer, I'd be happy to do that. We recognize that this is a platform, that is an instrument which ideally, because it has two very critical diagnostic technologies, could do a wide range of types of diagnosis. We had to focus on one for starters, so we picked fever-causing pathogens. The concept of someone comes in with a high fever, and the question is, what is actually causing the high fever? Certain of these diagnoses are difficult at an early stage, and having a biological sample would help you enormously in doing that. Is this the perfect list? No. This is a challenging list. This has RNA viruses, which means if we want to do nucleic acids, we've got to do RT-PCR. It has eukaryotic parasite, that being malaria, so we go after DNA and antigens for malaria, and to bacteria, so we have DNA from the bacteria. So we're going after RNA and DNA. In the case of malaria, we're going after antigens from the pathogen itself. In the case of everything else, we're looking at IgM levels. Why IgM levels? Well, it turns out that if you're trying to diagnose an early onset disease, and this is data from Swedish travelers who got dengue fever during their travels, and looking at the onset of the disease and what things they could find about things in the blood. So they're looking at PCR signals, that is the indication that there was actually nucleic acid in the bloodstream, and that positive PCR signal is the set of dots here. Notice at day 0, the positive PCR is the squares. It starts out high because they found that, and then it drops off by 5 days into the actual symptoms, whereas the IgM levels are coming up and peaking, in this case, at around day 5. So, in fact, if you started here, you'd want to look at the PCR data. If you started over here, there isn't likely to be a PCR signal, but there'll be an IgM signal. So by having both, we can not only find out they've got something, but we can even track the early stages of disease. So having the two of these things together, we think is really significant from a clinical point of view. So we would want to use this thing at a variety of points in the process as the IgM titer, as the bacteria or viremia peak and the IgM titers go up. We're not at this point planning on looking at IgG titers. We might in the future. The problem with IgG titers, of course, is they just tell you that someone had the disease. They don't tell you that they have it now. So we actually have to distinguish between IgG and IgM by knocking out the IgGs in the samples. So the idea is finger stick sample, a couple of drops of blood, add those into the disposable card, insert the card in the reader. After those pink things are done, no more user intervention until report the presence of pathogens comes out at the end 15 minutes later. Everything else should be done completely on board, completely automatically. And that's part of the challenge, is to automate all these things and to split the samples into the two separate processing streams for the nucleic acid samples and the immunoassays as well. Two optical methods. Why? Because we're optical types in my lab and other places analyze. So absorbance imaging for spots on particular sites for the immunoassays, and fluorescence intensity measurements, not necessarily imaging on the other side. So who's the team? It's not being done in my laboratory, please. I'm doing a piece of it. We're doing immunoassay development, but not even all the immunoassay development. Pat Staten's working on molecular systems. Nanogen is working on nucleic acid development itself. Micronics is working on the lab card and reader development and systems integration, and they're working with a subcontractor in Australia called Invitech, which is working on box design. And PATH, very important partner, PATH is working on assay validation. UNA stands for User Needs Assessment, a really critical portion of this, and as well as some systems testing. Let me briefly tell you about PATH, because most people know that the Bill and Melinda Gates Foundation's in Seattle, and that's helpful to say the least. But so's PATH, and PATH has been there for about 30 years. A 30-year track record of doing some rather remarkable things. They're a private nonprofit. They do medical technology for low-resource settings and have been doing that for 30 years. And that's always been a challenge, and their real secret is learning how to do it well. And we bow to them in terms of how to go about this. And having them on this project was absolutely critical to even the concept of maybe getting something out into the periphery. They have lots of funding sources. They've got 500 people worldwide. I think 350 or 400 of them are now in the Seattle area, but they also have these sites all over the world. So they have actual employees of PATH who are at all those dots on the map. So they have basically points of contact in most of the developing world, with the possible exception of Latin America, where they don't, at this point, have a lot of permanent sites. They've done a lot of tech transfer. At least 63 technologies transferred to commercial partners, and their route is to transfer to commercial partners, because only when you do that do you actually develop a product that goes out to people at the end of the system. Public and private partners of a variety of sorts, and again, frequently they've had funding from private donors like the Bill and Melinda Gates Foundation. They've done a lot of technologies, including vaccines. They're involved in some of those vaccine development studies we've heard about earlier yesterday. Delivery technologies, as well as diagnostics and contraceptives. And this is just some of their diagnostics portfolio. I don't want to go through all the all the acronyms here, but they've done a lot of commercialized technologies, most of them extremely simple. And that's part of the interesting point, is they've been working on dipstick and spot tests. Things are as exquisitely simple and inexpensive as they could possibly be. And what they've concluded is that the actual usefulness of those tests depends to a certain extent on userability at the far end. The question was, what could they do to improve accuracy and precision of those tests? And they made internally the decision that microfluidics might be a good way to go if it could be kept inexpensive. So there was a convergence of their thinking from their long-term work on diagnostics, our interest in microfluidics. At about the same time, the Gates Foundation came up with some funding to do this. So it was a nice accident. This is just an example of some of the PATH technologies. They worked a lot on lateral flow strip assays, which are a very simple technology, a really elegant one for immunoassays. And they've transferred all these. This is the disease and the manufacturer currently working on these. Notice that many of these companies are in India. They've done a lot of very successful work with Indian companies, where then they can actually knock the production costs down low enough that the people at the end actually get to use these things. And as I said, they're all commercial products. So on this project, their responsibilities are the user needs assessment and working on organisms for us, as well as validating assays. And I don't want to go through this in great detail, but just to give you a hint that, well, the first thing is user needs assessment. And what do we mean by that? Well, like I said, we're techies. So that we're very likely as techies to make a marvelous mousetrap for a place for which there are no mice. So the first question is, what's the rodent population like? What are the real issues that you have on the ground? And they have a lot of experience with this. And we were going to 3 places to do this. The first one was, in fact, India. So let me tell you about the India study. They spent time basically developing with consultants questionnaires and a whole process of interviewing people to discuss the general idea of diagnosis in this area, what kind of technology you need. And then they went to various sites within India, which, as you know, has a very wide range of economic situations. It's got people like, this is one of the AIIMS hospitals, which is a really delightful place that you and I would be perfectly comfortable going in and having surgery. And ranges to rural clinics. This is one about 75 miles out of Delhi, where the AIIMS hospital was. And what you don't see here is, if you look to the left, there's the medical waste disposal pit, which is an open area free for people to walk through and gather up whatever wasn't successfully burned in the previous attempt. So it's all the needles, all the sharps, all the biohazard stuff is literally sitting out in the open there. This is one picture of some of the laboratory conditions we're talking about. So those of us who work in labs in the U.S. go, oh, this is going to be an interesting environment. This is a reasonably well-equipped laboratory. This is a relatively centralized rural one. This is not the full peripheral end of the healthcare system. This is the next level beyond that. The good news is they actually have power periodically. They actually have things like a microscope and a centrifuge. That's very high tech. Underneath the desk, there's a couple of batteries which they use when the power is off, which they recharge. They switch back and forth to AC, DC. I think they've got an inverter on the wall over there. So they're making do, but you can tell they don't have a lot of money for the technical stuff here. So what we're talking about is fitting, at least initially, into a lab like this. We would add a whole range of capabilities they don't currently have. You also have to consider the courier supply service, how things get to the end of the peripheral system. And in India, this is a very typical way to get things out to the periphery. It's some guy on a horse cart or a bike, and clearly refrigeration is not an option. So people have heard about how you get vaccines out to the end. You have to be able to deal with things that have to be stored at 40 degrees C for a year. And that's sort of the standard. So we're doing an awful lot of testing in my lab right now of getting the chemistry onto these cards and then making it survive those sort of what Fran Liegler and I will know as mil-spec conditions. This is absolutely a requirement. If you could do it for the military, you might be able to do it for global health. But it's exactly the same set of criteria for storage and portability. So we've looked at a variety of things like what are the preferred assays, other assay types to consider, which ones do they like and don't like. Presentation of results is a really big question. Do they want a lot of detailed numbers? They just want to say, yep, it's malaria. And that's really a critical issue. Some healthcare workers want the detailed information. Others think of this as an example to work at what I'll call Western laboratory standards, and they want all the detailed information possible. So these are different clients, and right now we're sorting out exactly which way to go for which client population is the most important one for this point for the purposes of the grant. Willingness to pay is a big deal. We've looked at numbers, and that's obviously a very critical issue, and it's fascinating. And India is a very interesting place because unlike certain African countries, in India, most people get their health care from private providers, not from the public providers themselves. And they'd almost prefer to get them from private providers. So you're dealing with a couple of different economic approaches to how you distribute the technology. So we finished the India assessment. Brazil assessment's ongoing, and we're going to do a sub-Saharan country. We meet Friday afternoon to decide which sub-Saharan African country. So we'll accept votes until Friday morning. PATH is also working in immunoassay development. We're looking a lot at serology assays, and we've actually compared two different approaches. We're going to go with the indirect IgM capture assay at the moment. For those of you who worry about the technical details, the interesting challenge is that you're putting the antigen for the IgM down on the surface, which means if each different antigen can have a different ability to bind to your surface. And we'll talk about how we're doing that later. The assay validation's going quite well. Most are completed. There are a couple that are still in progress. The influenza one is we're still working on. We heard yesterday that in the cases of HIV infection, H5N1 infections, you do get viremia. It turns out is relatively unusual. So the biggest challenge in a way is looking for influenza in the blood, except in the cases of H5N1. Nanogen's working on reagent development for the nucleic acids. They've been developing all the probes. They have a very wide collection. This part of Nanogen used to be called Epic Biosciences in Seattle. Nanogen acquired them about, 6 months before we got the grant funded, which was another one of the interesting challenges when one of your partners suddenly changes hats. That was very interesting. So they're working on all the technology for developing the probes and primers to get the assays working for the PCR. They have a lot of existing proprietary technology that they can use, as well as non-proprietary sequence information that we're using for developing the reagents. This is just an example of real-time PCR for Salmonella typhi. This is a couple of biplexed assays because we put positive controls inside the system, so you're running two fluorophores simultaneously. As it turns out, we're not using real-time PCR for a wide range of reasons. It's difficult to implement, particularly in a cheap disposable. We're actually spending all of our detection time after the amplification's gone ahead and looking effectively at melt curves. And that's what we're going to do, which is obviously the primary detection method for identifying which one you've got. And that's where we're going to spend all of our energies for the detection. This is just showing, again, a triplexed assay, which is the influenza A and influenza B, as well as a positive control inside the system. And that works quite well. The assays are worked out. And the assays actually have been transferred to Micronics, which I'll show in a second. Pat Staten is how do I best describe, molecular engineer who's working in our department down the hall from us. And Pat's been working for years in developing what I'll call nano-related technologies related to polymer sciences, which give smart polymers interacting with proteins, nanoparticle systems that aggregate and move around in the presence of E-field or magnetic fields, but only when they've been aggregated to particular pHs. And we're developing a variety of smart polymer technologies for improving storage and release and capture, 3 sets of molecular functions. And one of his technologies ended up on the cover of Lab on its ship, so it's actually crossed over into the microfluidic world more or less efficiently. This is an example of a technology where you combine with magnetic nanoparticles streptavidin, biotin antibodies, and pegylation and as well as NIPAM, which is a polymer which precipitates out of solution at high temperatures or at, in this case, low pH, which allows you to cause particles to aggregate and become, only when they've been aggregated, pulled out of a stream and pull down their targets to a side of a channel. And it's one of the technologies we're examining. My group's working on immunoassays, and our primary task was to come up with the immunoassay technologies. One of the things you've heard that I worked at Naval Research Lab, so we're going to be here with Fran, as well as John McDevitt. John and I were also PIs on a salivary diagnostics project. It's wonderful being before these two guys because that way they don't get to steal my thunder by being first. And John's done some really exciting work on the use of porous matrices for getting effective concentration of analytes as well as really interesting sensitivities. And we said, that's a good approach. I won't say what happened before we made that decision, but let's just say that it's been an exciting 18 months we've been working on the project. But we're using porous polymeric membranes that are easily commercially available at this point and incorporating them into devices and then looking at, in this case, this is a sandwich immunoassay, where you then use an undetectable substrate, which you convert to a detectable substrate, and we're using the HRP TMB system for those of you who are aficionados. This is roughly what it does. This is a model system we use on a 96 well plate with a vacuum manifold and suck liquids through there. The membrane is a porous material. It has a surface, and what you do basically is you first decorate that surface with some capture molecule. And in the case of malaria, we actually put a capture antibody on the surface. We then flow the sample through and pull out, in this case, malarial antigens, which get stuck to selected antibodies. You then flow through a secondary antibody, which has on it a label. The label, in turn, is capable of creating or interacting with a substrate, which is uncolored to begin with. When it hits the enzyme, it produces an insoluble precipitating material, which is dark blue, which turns your membrane dark blue. This is a conventional ELISA technology, except that usually people turn the solution blue, and this is sort of mixing that with a technique usually used for histology. Gives you a dark blue spot. And like I said, in the 96 well plate, many dark blue spots. We've worked with a variety of membranes. So far, one of the best ones, the Paul Ultrabind membrane, which is an aldehyde-modified nylon membrane, as it turns out. And we get a wide range of responses over a wide range of concentrations. We've worked extensively with this material, and we think we've nailed the immunoassays for malaria. It turns out the other ones are a little trickier, the serology assays, because each one is different. And fortunately, it's a 5-year grant. Let's just leave it at that. We're using piezoelectric spotting method to print these things down on the membranes. We sort of cobbled together a commercial system with a whole bunch of stuff we have to build ourselves and run everything in LabVIEW so we can actually manipulate stuff. This is the jets themselves down here that we fire onto a small stage with an XYZ translator. Obviously, this is not a production facility, but what we love about it is inkjet printing is very easily scalable to something where you can make it dirt cheap. And that was a critical issue, was that we would be limited hopefully only by the cost of the reagents themselves, and we're using nanoliters, so we're not going to use a lot on each spot. This is an example of what the inkjet itself produces. This particular one produces a 15 picoliter droplet. That's overkill. We typically, these days, are shooting 500 to 1,000 of those down, so we can actually go to a coarser print system later. This is an example of the smallest spots we can easily produce, but this is just one colored liquid. This, on the other hand, is the actual amplification spots for number of droplets, and you can see there is a concentration dependence on the number of HRP molecules that go down there, but it actually works very well, and it's very reproducible, so we're pretty happy with it. We've reduced a lot of this to the polymeric laminate card systems that Micronics makes. This is literally the first run through this thing. We went, oh my God, and that was 6 months ago. So we've had 6 months of improvements on that. The cards themselves look reasonably elaborate. They are, but the beauty is that all these things can be compacted into small spaces. We ultimately think we'll probably have 2 separate membranes on 2 locations, one for the IgM assays and one for the antigen assays for this particular blood card, which makes things more complicated, but the good news is most of the plumbing and the samples can be identical. It's the fact that we have to do a dilution on the card in order to get from one level for the one set of antigens to the others. Micronics on the project is basically the systems integrator. They're putting all the pieces together, and they're the ones that are working with Nanogen to make the nucleic acid stuff work. And ultimately, they've got to have this last topic here, commercializing it in the developing world. That's actually, this isn't just an R program, it's an R and D and C program, which is a major challenge. And let me say, there was a huge amount of paperwork to get this thing put together for the Gates Foundation. Micronics makes commercial instruments now that do microfluidics. They built thermal cycling systems as well, and then monitor these things with nice sort of breadboardy, ugly stuff that you'd recognize as laboratory equipment. The cards themselves look somewhat cleaner in various stages of development at this stage for the PCR assays. They've now been reduced to practice to a version that we ship down to Australia, where they actually are working on integrating these things into the box. That should be done in about 13 months. We expect to have a fully functional prototype actually on the table. So if you really want to see how these guys are doing, come see us in 13 months. But we have a very busy 13 months ahead of us to get it done. This is just a demonstration of Salmonella. 35 cycles of on-card amplification. The PCR works fine on the cards. Right at the moment, we're shooting at about 8.5 minutes for the PCR cycling itself. So we can get all the PCR done in 8.5 minutes. There's a reverse transcriptase stage for the RNA ones at the beginning, and then we actually have to do the melt curves at the end. The whole thing adds up to maybe 20-something minutes right now. So entire process from sample to result, if it's going to be limited, as we suspect, by the PCR part, it's going to be about 20 minutes. We should have the immunoassays done in much less time than that. Should one ultimately design a card that could be used just for immunoassays where you didn't care about the PCR, it'll actually work much faster on the device itself. Oh, and by the way, can I go back? Let's see. This is 10-copy detection on the card. It actually turns out we're doing even better on the card than we did on the laboratory version of this thing. So in fact, it's working pretty well. This is just dengue real-time PCR with optical detection, although we're not using real-time PCR in the final version. This is an example of one of the cards, and this just show this to show here's like, these are bladders, basically blister packs on the card, which can be essentially broken not by your finger, but actually done with gas pressure. And right now we're looking at 2-zone amplification and shuttling liquids back and forth between 2 different temperature zones, which turns out to be perfectly adequate for doing the PCR, and then shuttling it off into a detection zone where optics are applied to the card. All the stuff gets kept on the card. When it's done, toss it away. This is about twice the size of a credit card in its current version. They are also working on another ELISA application, which involves direct formation of spots on surfaces, and that's working down to low levels. We actually have 2 separate approaches for a bunch of good reasons right now, which we can talk about, have to do with reagent sensitivity. But their version involves a flat surface, and they're able to see very nice development of spots on a flat surface using the same TMB amplification system. They've reduced this to spots on a card, so they're actually now looking at arrays of 16 to 25 spots on a card developed simultaneously. They're using bellows on card to rinse things and do most of the stuff you do in a conventional 96 well plate version of the ELISA, but do it on a card automatically with no hands whatsoever. This is an example of the immunoassay card, including sample separation. We'll be focusing a lot this year on fine-tuning the sample preparation issues, which are tricky, and there are 2 of them in parallel. So, to summarize, where are we going with this thing? Where we're combining what we call classic, that is, ELISA, PCR technologies, and emerging platform technologies, the microfluidic stuff, to perform multiplex assays directly from one biological sample. In a sense, we're borrowing existing technology whenever possible and trying not to invent new technology as we go forward. It turns out that's really a bad thing for a variety of reasons. The primary one is cost. If you want to actually get the lowest cost device, you don't want to use proprietary stuff you have to beg and borrow from some major pharma company. It's just a bad strategy. So we're keeping it as simple and as stripped down as possible. Because of the user needs assessment, we believe that our solutions will be responsive to the resource-limited settings. We're working closely with the Gates Foundation to make sure that that actually occurs. The initial target is this febrile illness panel, looking at fevers. We intentionally avoided AIDS, and we intentionally avoided TB, simply because the market is crowded for those. We wanted to go with something that, one, was different, number two, didn't have a lot of other people trying to trample us in their path. However, if the platform works, it is a relatively straightforward idea to simply switch the reagents on the card. If need, change the configuration of the channels, but ideally just switch the reagents and make it useful for a wide range of other samples and other types of diseases. And certainly, ultimately, HIV and TB will be ones we want to go for, but not in the first round. This is just my research group. This is just to give you some idea of numbers of people involved. Multiply this by about 5, and you'll get sort of the full size of the crew working on this project at various sources. Sounds like a lot of money. You distribute it that number of ways, and it doesn't seem like nearly enough or nearly enough time. So let's just say that the grand challenge is definitely something that we think of a lot. So I want to thank very much the Bill and Melinda Gates Foundation for an extraordinary opportunity to sort of go from being a techie to somebody who can actually make a difference. It's one of those opportunities you don't get many times in your life, and you know, it's exciting. And when it's all over, if we're successful, it's going to be very rewarding. In the meantime, I'm going to lose what's left of my gray hair. Thank you very much. Don.
Paul, have you had a chance yet to look at the sensitivities of the integrated
The integrated systems, the sensitivities, they're seeing down to 10 copies for the PCR easily for all the PCR assays on card. Each one of them has been validated on the card. We're actually now doing selectivity panels and doing a variety of things of tweaking the particular primers and probes that we're using for the system. But it's looking as if we don't have any sensitivity problems with the PCR at all. The immunoassay sensitivities are excellent. It turns out, and this is sort of stunning in certain ways, that one of the limiting factors so far has been actually learning what the clinical values are for some of the antigens we're looking for, particularly for the malarial antigens. So we're actually doing prospective studies in Kisumu. It turns out there's a major site in Kisumu that I believe is Walter Reed is the primary site there, and we're actually working with the folks at Walter Reed to get sites from there. We're actually getting samples from Vietnam, and again, prospective collection of samples to get influenza data. And so we're actually collecting stuff from several spots right now. Data collection begins, I believe, June 1. We're getting sample collectors prepared to go out there and then bring those back. All the testing will actually be done in the U.S. initially, and I think there's a lot of good reasons. I think the Gates Foundation, quite logically, doesn't want to be embarrassed if it doesn't work. They don't want to send it out until they know that we have a product, because we don't want to go approach people in developing world countries with a half-baked product. We want something that actually works well. The immunoassay sensitivities, I think, are superb with the HRP. We actually have a backup approach. I will tell you that one of the most critical issues that we're dealing with right now is it turns out that HRP is a wonderful reagent, except it doesn't preserve terribly well. It's one of those things you normally buy fresh from the supplier. So we are banging our heads right up against this issue of the antibodies, I will say, are relatively easy to preserve. They're relatively tough molecules, even though they're all a little different. HRP is the most tough one right now. So we have a backup plan, which we like very much. We won't go to the backup plan for about another 2 months, because right now we're doing extended testing at 45 degrees C of the reagents. Yeah. Please.
Thank you. Have you done extensive validation of some of these gadgets on the field?
Oh, God, no.
The febrile panel.
No. The validation in the field will only come if the instrument is validated in the laboratory. So the first thing we have to do in order to validate the system is to, one, prove that all the assays work in buffer, then they all work in whole blood. So we'll take whole blood, spike them with the pathogens as possible. We will also then collect samples, which will be stored and brought to the U.S., and we'll run the tests there. And if and only if it holds up at that point, we will then go to the Gates Foundation and suggest the possibility of doing trials on site. We don't want to waste anybody's time with something that doesn't work. I think that's the logic, because people have a lot of work to do, and we don't want to interfere with that until we know it's actually functional.
And you have an idea as to the price? You did mention
Let me explain.
that it should be cheap.
PATH has very successfully taken technologies, most of which have been simpler than this technology. Things like the monojected uniject needles that are now being distributed, for example, by Becton Dickinson, were actually developed at PATH. They have worked on a lot of vaccines. If you go and look at those little black circles that indicate cold chain stuff, that technology was developed at PATH. So they have a good history of working with manufacturers, both in the developed and the developing world, to come up with things that would be at cost. The Gates Foundation is clearly very interested in sustainability, and I believe firmly that the long-term goal is to have manufacturing for these things to occur in the countries where they are used. Now, I know that initially we'll probably work in countries like India and China because they have low-cost manufacturing capabilities. Brazil is definitely another option. This is very actively being discussed right now, is exact costing for these things. What the right price is going to be determined on the ground, and that's why we have these 3 user needs assessments. So India's done. It's not yet clear which is going to be the sub-Saharan African country we're going to go to. It turns out the Gates Foundation has 2 separate subcontracts to entities that are only looking at user needs assessments for diagnostics broadly. We're a subset because we're going after a particular type of diagnostics. We're going to piggyback on their work, and we're trying to coordinate which is that 3rd country, where we'll have 3 separate data points within a particular sub-Saharan African country to get, should I say, fine-tune the user needs assessments. It's obviously, in many ways, the most critical aspect of the project. And you'd say, well, why didn't you do it first? It's a good question. The problem was you couldn't really ask, what did you need, until you at least had something to propose as an object. So this is one of these things where we're doing both simultaneously. It's not ideal, but it's the way to go.
One more question, and I can move on.
What level of multiplexing are you trying to achieve and how?
It's a very good question. We're not aiming at 1,000 spots, 1,000 different tests at once. This is always going to be symptomatically aimed. Now, why? Size, footprint, complexity. We're not aiming at doing the sort of thing that you've been doing, for example, in sort of typing particular things. That's not our aim. It's to find a particular pathogen, particular condition, and as rapidly as possible and as cheaply as possible say it's there, it isn't there. In fact, right now, we're not even interested in how much of it's there. Ultimately, we'd very much like to do that, and we're building in the capabilities to determine what the levels are, certainly in the immunoassay side. Okay, that's going to be fully quantitative, but it may be that initially you can't see that from the outside of the box. It's a yes-no answer. We'll have to determine what the critical thresholds are that determine whether or not you report something, because we don't want to confuse people at the other end and give them these sort of graded answers. That's a very critical technological accomplishment. Could we go to much higher densities? We're aiming at the moment to build it off literally a cell phone camera. 2 megapixels, something like that. You can get a lot of spots in 2 megapixels, but obviously the smaller the pixel size, the smaller the number of spots you cover, the lower the resolution you're going to have in terms of the quantitative data. We're aiming right, and clearly we need to do multiples of every test. So we're aiming currently for sort of 6 to 10 reagents per side. Could you do a different set on the immunoassay and the nucleic acid side? Yes. But I don't think we're ever going to do more than 10 to 12, because the aim is to go after symptoms. Somebody walks in with a raging fever, terrible cough, bleeding sore, whatever it is, you're going to say, this is within these 3 or 4 things. What does the clinician want to know? He doesn't want to know genotyping. This is aimed at the clinicians, the people who are dealing with the healthcare on the ground, not as a research tool. That's a whole other product that we're not really working on at all. Okay. Thank you.
We're going to move on now to our panel on point-of-care diagnostics. And to moderate the panel and introduce our panelists, I'd like to bring down Professor Jung-Hae Soo, who is an assistant professor in the Department of Bioengineering, interested in bio-inspired nanoscale diagnostics and therapeutics. Dr. Soo.
Good morning. To carry on with the very exciting discussion on point-of-care diagnostics, we have three panelists this morning. First is Dr. Rebecca Drezek. She joined Rice Bioengineering in 2003, and already she's Associate Professor of Bioengineering, so that was 3 years, and also Associate Professor of Electrical Engineering. Her research focus is on the development of optical imaging technologies for biomedical imaging applications. She has a tremendous list of awards that she's gotten, and including she was named as one of the top 100 young investigators from the MIT Technology Review. She has the Beckman Young Investigator Award, as well as a Culture Foundation Early Career Translational Research Award. And most recently, she has received a $3 million grant for her work on breast cancer. She has been named the Era of Hope Scholar. And she has over 50 published and in-press papers and 6 patents and patent applications. Next, we have Dr. John McDevitt. He joined UT Austin as an assistant professor in chemistry in 1989. He has been there since, and he's now the full professor of chemistry and biochemistry. His research focus is on developing new biochips, most recently on saliva-based diagnostics. He has gotten several awards, including the Presidential Young Investigator Award, as well as the Gates Grant for HIV monitoring biochips for use in Africa. He has over 140 publications, over 95 patents and patent applications, which is one of the largest patent portfolios in UT history. He just started a new company called LabNow for developing microchip-based sensors for use in resource-poor settings. And our last panelist is Dr. Frances Ligler. She is the Navy Senior Scientist for Biosensors and Biomaterials. She's also the member of the Bioengineering section of the National Academy of Engineering. Her research focus is on biosensors, microfluidics, and nanotechnology. She has received the Homeland Security Award by Christopher Columbus Foundation, as well as the Presidential Rank of Distinguished Career Professionals by President Bush. She has over 300 publications and 24 patents, and collectively, her publications and patents have been cited over 4,000 times. So I'm very happy to welcome the panelists this morning.
So I appreciate the opportunity to speak here this morning, and really what I want to do over the next 15 minutes or so is talk about what I see as some of the opportunities for Rice in particular in the area of point-of-care diagnostics. What do you say?
It's on the...
This? Sorry. Oh. Okay. So what I'm going to be focusing on essentially is photonics-based technologies. And really what you heard in the last presentation was an example of a lab-on-a-chip-based technology. And here what I want to do is talk about techniques that can be used for things like urine, saliva, blood, that you could use in a lab-on-a-chip type setting, but also more focus on strategies that you can use when you want to image tissue in vivo without having to necessarily remove any sample from the body. And so to do that, I want to talk about basically 2 different areas. And so I want to talk about in vivo optical imaging strategies, both that are spectroscopic-based and imaging-based, where we use different forms of endogenous signals to give us contrast. So fluorescence, where we can see various biochemical markers, scatter, where we can see morphology. And then I want to talk about how we can couple miniaturized optical imaging devices with nanoscale agents, which allow us to see other kinds of activity that we might be interested in, which is overexpressed surface markers. I'll give an example of enzymatic activity, and then really genetic expression, imaging mRNA and live samples. And finally, I'm going to concentrate on the areas that I think we have something to contribute to at Rice, and there's a lot to make these kinds of techniques more viable, a lot of other kinds of technologies that we are not as well equipped to, and so we need to collaborate with others to make that happen. I want to talk about two examples of collaborations. One, an engineering research center, which is in its first year that Rice is a part of, and one, a proposal that Rice recently put together with the speaker who will follow me, and several different institutions in Africa in the area of point-of-care diagnostics. And so really Rice has what I see as three main strengths that contribute to this area. And so the first is really essentially the areas of optical imaging and nanotechnology, and these two are really well integrated already in what we have going on at Rice. And the third is signal processing, wireless, low power. We have a number of people in the electrical engineering department of tremendous strength in these areas, not as well integrated yet, but really have a lot to contribute if we're to develop these kinds of techniques in a meaningful way. And the perspective that I'm going to come at this from is essentially optical molecular imaging. This is what our lab does. So we work somewhere in between photonics, nanotechnology, and medicine, developing pretty cheap point-of-care devices that can be used for screening, detecting, and monitoring disease. And we do this starting from what I would call a basic science perspective, in that we use a lot of computational modeling, a lot of fairly fundamental experimental measurements to guide the design of devices, which we then take into clinical trials. And in our lab, we've really focused in the past so far on the area of cancer. And so this is similar to some of the charts we saw yesterday. And really what this plots is the mortality rates over the past 70 years. And so what you see in most of these cases is really that there hasn't been a lot of change. And so the work in our lab, the work in Rebecca's lab, and many other labs on campus has really been focused on technologies that we could use for early detection. But a lot of these same kinds of techniques can be adapted in various ways for other kinds of infectious diseases, and I think that's one of the directions we're really interested in heading. And so the reason we're interested in these kinds of strategies, optical technologies are getting progressively more inexpensive. I'll try to show you some examples of how something that might have cost 4 or 5 digits not too many years ago you can do for under $100 today. Very portable, very easy to use, very automated type techniques, that what you measure depends on how you're looking at these kinds of signals. You can look at endogenous signals where we measure essentially morphology or biochemistry. We look at things like hemoglobin, like metabolic indicators, like changes in collagen crosslinks, all of which tell us something about the pathology of the disease that we're trying to detect. And then we can couple with imaging agents to look at other molecular features that we might be interested in. And so I want to fly through a bunch of different examples of these kinds of techniques. And so first I want to talk about spectroscopy and imaging, and really you can do this in sort of two kinds of forms. You can think of a spectroscopy techniques where what you're using, what you're holding, is something that looks like this, a little pen, a wand, and you're getting this wavelength-dependent intensity information that you're using for some kind of diagnosis. You're not getting a picture per se. And the other form, and so we measure one spot at a time. If we want to cover more of the tissue, we can develop that into an imaging form, but now we're going to have to pick particularly what wavelengths we're most interested in looking at. And these are both examples from ovarian cancer. And so we've had 2 ovarian cancer trials going on in our lab, one at MD Anderson Cancer Center, one joint at Arizona Cancer Center. And so just to give an example of what these technologies, kind of the path that you go through, so you start with some kind of design process. This is an example of what the probe is. You can see the finger there, you can see the side of it, and there are going to be fibers that the light is coming in and out of. And so you go through a process where you collect a lot of these curves that give you information. And so this is from a study that had about 100 sites. And so you can see there's differences clearly in what the normal tissue, what the cancerous tissue is looking like, but how do you go back and come up with a diagnostic estimate based on these? And so then you can go through a variety of strategies. You may try to extract various kinds of biophysical parameters that are telling you something about the process. You may forget that entirely, just use statistics based on the differences in these curves and come up with some kind of cross-validated algorithm. And you can get accuracies that are quite good. And one of the things I would point out from this study is if you look at, these are some of the wavelengths where we did the best. These are wavelengths now where you can use much cheaper devices. So you see this blue light here. The light source that's being used in this device is about a 4-digit price tag. This is students in my class for the fall who are essentially making a super cheap LED little flashlight for oral cancer detection. The LED they're using here and this light source is the LED is maybe 65 cents. The whole light source is less than $10. So these kinds of technologies that were maybe a $30,000 device not so long ago, really you can do much, much more cheaply today. Now, can we improve further on where we are? I think there's still a lot more to go. And just to give you an example of that, what do we show here? This is the same patient. We have a normal site and abnormal site. And so the normal is the blue, the abnormal is the red. This is the same sites. And what you see, the only difference between these two curves, you can see in one case the normal curve is higher and the other is the abnormal, but we're really measuring the same patient is the depth at which we're doing that fluorescence measurement. So as you change depth in the tissue, you're measuring different types of features. And so how do we know? And so when we try to separate out the epithelial signal that we're particularly interested in from the stromal signal, we can't really prove that's what we're doing here, but it's what we think we want to do. But we can go back and do phantoms, which let us show what's really going on, because we make something where we know exactly the optical properties of each layer. And so we ask, can we really isolate this epithelial signal we're interested in from everything underneath in the tissue? And so to do that, basically what we do is we make these phantoms, and in the lower level, we change the optical properties of the stroma really fairly dramatically, and we try to look for these wiggles in the epithelial layer. And so in these two cases, and you see sort of different periodicities of the wiggles, no matter what was going on in the bottom layer, the epithelial layer looks the same. And so there's a few people who are at optics in here, so I'm just kind of highlighting one little bit of technical detail on what we're doing there. And so that's basically a spectroscopy example. What if we want to do something that's imaging? What if we want to switch our contrast to scatter? And so let me give you a few examples of technologies like that. And so this is reflectance confocal microscopy. And so here the nuclei that you're seeing is essentially what's giving you your signal. So you get a picture that looks very much like what a pathologist would use to form a diagnosis today. If you want to image deeper than you can do using reflectance confocal, you can use a technique called optical coherence tomography, a little bit of an optical analog to ultrasound, where you map out the backscattered reflectivity as a function of depth within the tissue, get about an order of magnitude increase in resolution over what you would get with high-frequency ultrasound. In these cases, we're using just the endogenous scatter. We're not putting anything into the tissue to get our contrast. Now, if we want to change, so say instead of wanting to look at the nuclei, so we want to look at the HER2 expression in this sample. And so what we're going to do is turn down the light level way down. So now the endogenous signal just looks black. And so now we have a picture that looks somewhat different, because the inside of the cells look dark, and we're seeing this sort of web around the outside of the cells, and we're using nanoparticles that scatter, that are basically targeted to that marker that we are looking at. You can do the same kind of thing in OCT, and this is just a tail vein of a mouse, and these nanoshells going in, and it's lit up, and this is a tumor that's being lit up by these same kind of imaging agents. And so I want to use this to switch into the second half of the talk, and how we can use different kinds of nanomaterials to look at different types of contrast that we might want to. And so the first example I'm going to talk about, and I'm going to try to emphasize examples where we can do some kind of dual integrated imaging and therapy, some kind of point of care, which ultimately we would, not right now, but we'd like to be able to use it in a point of care setting down the line, where you wouldn't need 2 different visits. Okay, so this example, we're going to start with some kind of non-conducting core, generally made of, in this case, silica, and we put a very thin metal shell of a particular thickness on here. And these are particles called nanoshells that were originally developed by Naomi Hallis, who's another faculty member here at Rice, and Jennifer West, another faculty member in the bioengineering department, and I have been collaborating on this project. And so basically the thickness of the shell relative to the size of the core is going to tune the optical properties of the particle. And so what you can see is a very small difference in the shell thickness. So this is a 15 nanometer difference in the shell thickness. You've switched this optical resonance 600 nanometers. And so basically, as you increase the core shell ratio, you redshift these particles. And so you can get optical properties in the near infrared region, which is most useful for imaging. And so I want to give a couple examples on this integrated imaging and therapy idea. And so what we're going to start with here are nanoshells that are going to be and this is cells that overexpress HER2. And so what we want to do is light up the cells that are expressing this marker, and then be able to kill the cells without killing surrounding normal cells. And so this is basically the in vitro example. And so this is the experimental case. And so this is just dark field microscopy. And so you're just seeing the scatter signal on the surface of the cells here. And this here is your laser spot for when you basically turned up the laser power. We designed these particles both to scatter light at one wavelength to absorb light. At another wavelength, we use the absorption to make them hot to just do direct ablation. And that's what's going on here. And you can see in the control cases that's not happening. Now, this is just a cell example. How does this translate to animals? So let me show you an animal model. And so now we're going to switch our imaging to OCT. And so what you can see here, and this is a tumor, these are just what normal OCT images look like, normal tissue tumor. And so you can really see the difference after you've added the nanoshells in. And so you can use this to find the right spot that you're going to do your treatment. And so now, and just to show you what it looks like, here's the mouse, here's our OCT probe for imaging, here's the laser for therapy, and this is a trial that we did with Jennifer's lab. And so at day 0 after treatment, this is a treatment time, everything is the same, and the animals in the control classes, of course, their tumors are continuing to grow. In this treatment class, the regression is pretty good. So this is just an example of a dual imaging therapy. That's an animal example. And one thing people often ask is, do you really need the imaging here? So I just wanted to give an example of what we're really using the imaging to do. So now we're starting to do these kinds of studies in Jennifer's lab with targeted nanoparticles. And we really need to understand the kinetics. When do we want to do the treatment? When do we have the best accumulation? And so here we're using OCT as we're doing the animals to monitor that. And this is the most simple automated way you can. We're basically just completely integrating the intensity into these images. And what you see is the time course with these VEGF nanoshells was much faster when we had the optimal delivery than and this is just 8 mice the first time this was tried, than with the pegylated nanoshells. So it's just an example of how you really use this imaging information to tell you something about when you want to turn on the laser for the therapy. And so I talked about these nanoshells, which are essentially a plasmonic particle. So we use basically the oscillation of electrons on the surface of those particles to give the property. And by tuning the size of the particle, you tune the frequency, sort of the way you tune the size of a stringed instrument and tune the sound you hear. Another type of particle that we're working with are basically quantum dots. And so here, instead, what we're doing is to tune the band gap, essentially to tune the color that we see. And here you see visible quantum dots. What we've been focusing on in our lab is more quantum dots where we can extend out the excitation and the emission wavelength really into the near infrared, because we want to be able to excite in the red. We want to be able to emit in the 800 plus nanometer region. You can sort of see why we want to be able to do that here. And so this is an example. Here are conventional quantum dots in the red. Here are near-infrared quantum dots. You basically, this is a tumor phantom model, and so you're going deeper, and you can really see the blur that you're getting because of the scatter when you're trying to image. And this is basically a mouse phantom model here, versus with these with the near IR dots, where we're able to image much deeper, really preserve that, and we can go fairly deep. And so this is what this is a centimeter, more than a centimeter deep. You get very nice linear optical properties with these particles. We're doing our surface coatings a little bit different. I'm not going to talk about that than other groups are for this. And so we're doing other things with quantum dots. This is an example of photo-switchable quantum dots where we encase in the polymer that coats them a photo switch so that we can change colors. And so basically we can, in this case, we're just switching between red and green, and you can't see the cells very well here, but this is just an example that you can do this in live cells and this kind of thing. So we're doing a lot of other kinds of materials work as well. Now, why do we care about quantum dots? One of the things they really are good for is for multiplexing. And so this is an example where we want to monitor enzymatic activity. And so here, what we are going to do is start with a quantum dot that's, and this is just a red emitting quantum dot. And so we're going to attach these nanoparticles, these gold particles around there that absorb light. And they're going to be connected with a degradable peptide. And so what's going to happen is when you have activity of the enzyme that you're interested in, they're going to get cleaved off. And so this quenched quantum dot is now going to become emissive. And really, this is just what this shows. And the time course, if you ask why the time course is so slow, is basically the temperature that we were doing this experiment at. We're doing it at room temperature for various reasons. And so let me just give one final example. And so the final example I want to talk about is, what if we want to image mRNA. And in particular, I want to talk about an siRNA example. So siRNA, there's a lot of kind of therapeutic work being done with these particles, and people have sort of attached the fluorophore to them. Can we see if it gets into the cell or not? Doesn't get into the cell. But there really wasn't a way to see, are these particles being, is this siRNA being activated? And so essentially what we're going to do here is create our own molecular beacon. And so we're going to start with the siRNA, and so we're going to put a fret pair essentially on the end of them here, and then we're going to make the loop of a molecular beacon with the PEG. And basically when you control the size of the PEG, you're going to control the kinetics of how this is working a little bit. And the way we get this to become a molecular beacon is essentially using the cell's own machinery. And so when this whole thing is going to incorporate into the RISC complex, it basically gets chopped, and we end up with a 9-base paramolecular probe. And then it goes on to work basically the way a normal one would work. So if we have the presence of mRNA, and I'm going to show you a telomerase example, the thing is going to open up, and we're going to see a different color change. And so in our cells that are negatives, that don't have telomerase, we're going to see yellow. In our cells that are, we're going to see green cells. And so this basically, and these are sort of the first experiments that we did, and these are different size loops and different time courses, but basically you can see in these living cells, you can see the presence of the telomerase by this green color. And most importantly, the question was, can we do this and still preserve the knockdown that you're trying to get in the first place with this RNAi? And so in fact, that seems to work. And so these are the knockdown, and these are the control expression. And so this was just really our first example of this concept, but it's just a little bit of a twist on a way you might develop probes for more genetic beacons. And so to finish, I just wanted to give examples of 2 sets of research collaborations that are really taking advantages of the strengths we have and the strengths that other institutions can provide. And so the first I wanted to talk about is an NSF Engineering Research Center that a bunch of faculty in science and engineering here are involved in. And this is headed out of Princeton, but 2 of the thrusts, the sensor systems thrust that I'm directing and the testbed thrust that Matt Fraser has been directed, are both headed out of Rice. And really what this ERC is all about is developing point-of-care miniaturized sensor systems that take advantage essentially of a lot of what has been going on in the development of quantum cascade lasers and some other kinds of laser technologies where you can do very tunable, very cheap source development. And so there's people working on sources, there's people working on detectors, on materials, on sensor systems, and then there are a variety of medical application test beds and environmental test beds that are ongoing. And this is now in its first year. And then I wanted to move into the next talk by talking about a proposal that isn't funded yet, but we hope it will be, that we submitted in February, that Rebecca Richards-Cordham is the PI on, that was really focused on point-of-care diagnostics for infectious disease and really integrating all of the different pieces. And so I talked a little bit about the optics side, the agent side here. I didn't really talk about the sample prep, the data analysis, the reporting, how you have all the other things you need to have a sensor system together for some very specific infectious disease applications. And so the other PI on this, who you're going to hear, is John McDevitt, who you're going to hear from next. And so I'll just stop by acknowledging all the people in my group who did all of this and the people who give us the money to do it. And finally, to thank Rebecca and Michelle. I think there are a whole bunch of us here who somehow bumped into them one day, and sort of the whole direction of which our life emerged changed afterwards. And so I'd be happy to answer any questions or for doing that.
We're going to do discussion.
Good morning. As my slides are being teed up here, I'd like to begin my talk also by thanking Rebecca for the kind invitation, Paul and Rebecca also for the introduction to the themes that I'll talk about today. My particular research group is focusing on the development of a series of lab-on-a-chip-based systems, and I'm going to take one piece of our research and drill down into that and focus on an initiative which we describe as a CD4 Africa initiative that targets HIV immune function measurements in point-of-care settings. Our goals for this program are stated here. We want to work towards developing the next generation of lab-in-a-chip systems that are affordable and accessible. And this is key. I think Paul had a very good introduction and emphasized the importance of making these systems accessible and affordable, and that is one goal that we share here also. Another thing that we want to develop in our system is the ability to rapidly go from one kind of test to the next kind of test, and especially in resource-poor settings and also here in the States, it's clear that cost is key, and if you need to spend 3 to 5 years and nearly $50 million each new diagnostic device you develop, it becomes cost prohibitive. But if we can leverage a constant platform like the microelectronics industry has, then we have the potential to make these systems affordable. Now, the last thing I want to emphasize is the need to get the chips out of the lab and get the lab on the chip. This term lab on the chip has an origin back 20 years ago. It's a very sexy descriptor to put the entire lab in a little bitty chip, but the actual execution, the integration, has been a significant challenge, and so that'll be one of the things that I talk about. Now, this particular effort on CD4 HIV immune function measurements has many different collaborators that are highlighted here, and this program has evolved through different phases. It started off with anthrax detection and bioterrorism in the kind of post office setting, and developing the conceptual basis for doing these kinds of tests at the University of Texas. We then began to collaborate with Bruce Walker and William Rodriguez at Harvard Mass General, and we adapted this spore detection system into a CD4 detection system. So I need to acknowledge their significant contributions in going forward here. More recently, we've begun to take the steps of getting this into the real world. A company called LabNow has been launched in Austin, Texas. Some of the key members of the company are shown here. And even more recently yet, we've begun to take steps to have these systems tested, not in the developer's lab at the University of Texas, but this is happening at the University of Kentucky, and you will see from us in the future further steps to doing these tests in Africa. Now, yesterday I think we got a good introduction to some of the issues related to the AIDS pandemic, and I won't spend a lot of time on this, but except to focus on one key issue here, and that is that while it's very encouraging that we have new developments to create a source of retroviral drugs, 90% of the market, the patients in Africa are underserved. They're not near these regional clinics, and we need to find a way to bring our facilities out from these labs and into these kinds of clinics. And if you look at what some of the bottlenecks are here, it's no longer the lack of availability of the antiretroviral drugs. It is stated in the June 2004 issue of Science, where they focused on AIDS problems in resource-poor settings, that the lack of an ability to measure CD4 counts, which is traceable to the immune function, is one of these key missing links. And so that is something that's done within the central lab, but not so readily out in more rural settings. Now, CD4 measurements provide us with a window and tell us information about the immune function of the patients. And after infection, there is a seroconversion. And what I want you to pay attention to is this particular line here. This is the CD4 positive T lymphocytes. And when this begins to head down south here and goes below 200, we enter the AIDS syndrome. And if we don't begin to apply treatment at this point, then the patient dies relatively quickly. So this is a key measurement that has to be done. In developed countries like the United States, the gold standard for making this measurement is a flow cytometry. It's been king in this area for 35 years. It's well established, it works well, but it's relatively expensive, it's a delicate instrument, it requires an expert user to set up and maintain the instrument. It's not mobile, and these are some of the issues that are problematic for doing the measurements in more rural settings. So in Africa, this is our lab. It's this kind of environment, and the challenge is that we really don't have a well-defined laboratory infrastructure for most of the situations. We have trouble keeping trained technicians that would run these kinds of measurements. There's few resources to set up an infrastructure. So we need to change the paradigm. The experiment has been done for 35 years with flow cytometry. It doesn't work. Something new has to come. Now, we've put together a platform, a system that's flexible, that can do both soluble tests, so protein antibodies, general chemistries, are done on bead-based systems that are shown up here. And I describe this as a chemical processing unit, or CPU, to use the electronics analogy. I'm not going to talk about that today, other than to mention that particular system fits on a credit card, the same format as the system that I will focus on, this system down here, which is a cellular processing unit. Both of these systems feed into the same analyzer unit, which is here. It's about the size of a toaster and weighs about 10 pounds and has all these actuators and optics built into it so that it can manipulate these biochips. Now, for the CD4 detection, we are making a very simple sample processing strategy, and it involves taking whole blood and sending it through a membrane system composed of a nuclear pore membrane. And we take the much more numerous red blood cells, direct them through these little bitty holes. The red blood cells have the biological function of going through nooks and crannies and capillaries, and so they work their way very effectively through 3-micron holes. And what does not exit through this trap in the floor are the white blood cells. And so we now have a static target, and that becomes part of our cellular processing unit. So this is an electron micrograph of one such system, and it gives you a little bit of mechanistic information about what's happening before this was imaged here. But here are the white blood cells, and you see the red blood cells that are making their way out. It's relatively rare that they get stuck at the door, but for the most part, we're able to process a sample quickly and efficiently. Now, the data analysis happens as I show here. So we bring in a fluorescent cocktail. So we basically spray paint these cells with fluorescent dyes, 2 different colors. And so the data analysis begins with snapping a picture with a CCD, following some of the things that Paul talked about, using inexpensive components to make these kinds of measurements. We begin with a snapping of a picture here. That's converted now to a grayscale image, and this is now converted to binary image. And you see at this point that the image is relatively messy. And there's regions up here where there are things like precipitate. Every measurement has its noise. This measurement does too. But with this imaging strategy, we can apply our knowledge of how big a white blood cell is, how big a T lymphocyte is, and very accurately pull out the different white blood cells as shown here. Okay, so this is now done in two colors. It's kind of hard to see here, but there's green on the left and red on the right, which pulls out CD4 surface marker on left, and then the CD3 on the right. And in order for this to be something that we care about, for adults, it's the CD4 positive T lymphocytes. That carries both of these markers. So you basically add the yellow and the green and get yellow. And it's as simple as going in and counting the yellow dots. That's basically our measurement. And this particular series of representative images shows data that was acquired from our Botswana trial done in collaboration with Bruce and Bill. And it shows 3 female patients. The one over here, you can see, you don't need to look necessarily up here, but the CD4 count extracted from our automated macro reads 83 in this case. This patient was on death's doorstep. And a little bit more stable in immune function is this patient with 271, but and a very healthy baby girl, an infant, is shown here. And again, you can see this simply by counting the number of yellow dots. Now, early on, we did this study in Botswana. We did a similar trial in Boston, and we published this work in PLOS Medicine. One graph that we extracted from that study is shown here. This is CD4 percentage. And the correlations are decent. They're on the order of what you would get with a flow cytometry. But this was done in a lab. And it had all the benefit of a white coat and a series of technicians. So it really did not make the step that we wanted to make. And this was featured in Science. I think the publicity was premature, to be honest. It puts us in a mixture of companies that actually do have products on the market, but I think the more important things have happened more recently when we've raised the bar even higher yet. And now the goal has been, and this has taken us the last 2 years to do this, but to get the chips out of the lab and to bring the lab on the chip. And this last piece is what I'll focus on in the remainder of my talk. Now, in the Botswana prototype, if you look underneath the hood, we had basically a microscope in a box. And we also had pipettes. We had the white coat. We had trained technicians. We had stable power. We had a refrigerator. We had access to transportation. Now, our goal is to create this, and we've done this now, created a series of prototypes that remove the infrastructure. And that's key. This is battery-powered system that we have here. We have stable reagents embedded into the cards. We no longer need the refrigerator, no longer need that, no longer need the venipuncture, no longer need the white coat, no longer need to make the volume measurements. All of this is happening within this chip. And this particular chip here, I think I have somewhere, but it's based on injection molded plastic systems, making this inexpensive. A clear version is shown up here. Now we're making these out of black plastics. This becomes easier to seal via laser sealing process. But this system is integrated. It eliminates the waste. It eliminates the pumps. We do this with a series of of liquid blister packs here. We no longer need the venipuncture. We need a drop of blood. The reagents are embedded in the solid state form. That allows us to get rid of the cold chain. We've shown that through accelerated stability studies. And ultimately, we're doing the function of a flow cytometer, putting all the pieces together here. Now, Mark Klein had a very nice talk yesterday about HIV pediatrics, and we've been concerned about not only treating adults, but also pediatric patients. And so as we've moved forward in building this miniaturized lab, one of the things that we've done relative to our first series of efforts is we've changed what we're looking at in a subtle way, but in an important way. And so now we're using these kinds of surface markers, CD4 and CD2 and CD19. We use two colors, but out of this imaging strategy, we're able to pull out four things, and that's key. Out of the yellow measurements here, we get the absolute count, which is traceable to the HIV immune function for adults. Out of the a ratio of yellow to red, we pull out the CD4 percentage because this combination of cells brings us all the lymphocytes, and that's what you need for infants. And then we also get the monocytes here and the total lymphocyte count. So we get a few extra pieces of information that can be clinically useful. Now, this particular movie shows this chip in operation. You saw an excess amount of blood initially go into that card. It wicked into the guts. The lab card is now being put into the analyzer. In the analyzer, there's a series of actuators that come down and at specific times synchronize the delivery of fluid. Now you're seeing the red blood be delivered. It's filtering the red blood cells. This continues on. Then the reagents come in, and this is not subtle. You can see when the reagents arrive. Okay, so the huge influx of fluorescent cocktail. This persists for about 3 minutes. It stains the cells at this point. Then we actuate the other blister, and we rinse away the excess reagent. Then we snap a series of pictures in red and green, and we do this image analysis algorithm that I've just referred to. So all the pieces are coming together nicely. This was studies that we did about a year ago, and more recently we've begun to do some clinical trials to see just how good the data is. This particular correlation study done on the lab and the chip, so the fully integrated system, we've made about 10,000 chips now at this stage. This 200-patient study looks at a correlation between the total lymphocyte count measured by our biochip over here versus a flow cytometer. Correlation coefficient quite decent, on the order of what you would get if you went one flow cytometer next to another. What you really care about, if you want to make the diagnostic decision, is the absolute count for CD4 at around 200. And again, this is challenging for flow cytometers. We get very respectable errors. As you see here, the 95% confidence interval is shown here. It's about 80, which is, again, quite good relative to the existing technologies. And this particular data graph shows CD4 percentage. So this is done with Timothy Shacker at the University of Minnesota, and it shows the efficacy of this approach for pediatric patients. I'm going to finish up my talk here just by giving you a little bit of perspective. Looking at the existing infrastructure, if patients are lucky enough to be close enough to a regional clinic to donate a sample, you have a series of weights that you have to deal with. And one is delivering that sample to the central lab, and this can take up to 6 weeks. And then you need to find the patient. If the CD4 counts are below 200 and treatment needs to begin, you need to go back and find that patient. And that's not always easy. And so it's days to weeks to months before treatment can be put into place. A point of care system, like the one I've just described, has the potential to be at the patient's side and within 10 minutes to provide the full diagnostic solution so therapy can begin during that same visit. Well, in finishing my talk, I won't necessarily go through all these details very carefully, but rather just leave this up for you to take a peek at. We now have an alternative to flow cytometry, and that's the key message. If you remember one thing from my talk, that there is an alternative to flow cytometry that is point of care, that is being scaled up in a manufacturing way as we go forward. It has a number of advantages relative to the flow cytometer and has the potential to be expanded to other kinds of diseases in the future. Thank you so much for your attention.
Again, thank you, Rebecca, for inviting me today. It's a real pleasure to be here. I'm here from the Naval Research Laboratory, which is basically a contract research house in the D.C. area. We're all on soft money, so we survive on grants and contracts, and I'll guarantee you that means there's no dead wood. We don't have time for it. But the way I have structured my research in the 21 years I've been there is to develop technologies of interest for making small automated systems for a wide variety of applications. And part of the reason for this is that although the DoD would like to see me focus entirely on military applications, the DoD won't pay to have those devices made. So if I'm going to go out and find a corporate partner to manufacture the technology that I think has the possibility to make a contribution, it has to have multiple uses. Primarily because the DoD is one of the flakiest customers in the country. So we try to do, and so as I go along, I'm going to give you a very quick survey of some of the systems that have come out of my lab. We've taken them from idea through prototyping, through field testing, and some of them on to commercialization, but clearly for multiple applications. So think about how you might use these for addressing global health problems as I go along. Which one works? Okay, these are some of the systems that have come out of my lab that I'll talk about a little bit. This one does saliva testing for drugs of abuse and won the runner-up for Best Clinical Device of the Year in, I think, 2002. We've put them on small automated planes and flown them through the air looking for toxins and pathogens. We have portable devices of a variety of kinds. Again, I'll talk to some of these. There must be a better way to do this. Oh, here. Okay, I got it. First one I'm going to talk about is now being the project manager on this technology is now Ann Kusterbach, and she's developing it mostly for unexploded ordnance, but it's the same technology we developed for drugs of abuse in saliva. We've used it for antibiotics in milk. We've used it for PCBs in groundwater. It's really good for low molecular weight molecules. After doing the immunology for 10 years, I came up with the basic idea for this system and then didn't realize till later it should never work. And the way it works is that you have a continuous flow going over antibodies immobilized on a solid support. And these antibodies have been saturated with a fluorescent analog of the molecule you want to detect. You put in your sample into this flow stream. If the sample may or may not contain target. If it does,
it displaces the fluorescent analog from the antibody, which goes downstream, and you can measure it. All of this takes about a minute, and it's very simple. You can add sample after sample after sample because you can never displace more than a few percent at a time. And so to the user, it looks reagentless. And so they can do 60, 80 different insertions, even of positive compounds, and get responses. The reason it should never work is that if the antibody binds that fluorescent analog so tightly that it doesn't wash off in 20 minutes, which is what the KDs of this antibodies tell you should happen, but it doesn't. I mean, it doesn't wash off that fast. If it doesn't, then how can you get a displacement within a few seconds when you add the real target? And what we think is happening is really a function of the laminar flow effect and keeping the fluorescent target right next to the surface. So instead of being diffusing out into the bulk solution and being washed off in 20 minutes, it stays right close to the surface and tends to rebind either to the same antibody or the one next to it. But when you have target in your sample, you can keep that rebinding event from happening and actually wash the fluorescent analog downstream. It took us, working very closely with Sina Rabbani at Hofstra, about 7 years and about 7 theoretical papers to figure that out, but eventually we got it. This shows a young lady from our lab working on a study that we did to detect explosives in groundwater at Superfund sites. The system was made up into something that looked like a CD player, so if you want to go to the field, you just put it in the case with your laptop and off you go. The antibodies, and we have actually patented the way of putting the fluorescent analog onto the antibodies and keeping it stable for over a year in storage before we used it. So that can be done. And we mapped plumes of the explosive going into the groundwater where it is toxic to people and animals, and showed that they matched up very closely with the standard laboratory techniques that would take 2 weeks to a month and cost $500 to $1,000 a sample. And it was also very, very sensitive. When we wanted to detect TNT in seawater, we actually made antibodies against TNT that would function in seawater. So seawater was a little more challenging than blood or milk or some of the other things we looked at. But we can do that too, and we're getting the equivalent of being able to detect a tablespoon in a swimming pool. And so we have, we're in the process, or Anne is in the process now of putting this on an underwater vehicle. This is the second underwater vehicle she's using that can just swim like a fish through the ocean and pick up plumes of explosives as they leak out of unexploded ordnance. But that same technology is also can be used for monitoring things that would contaminate groundwater, for instance, or for detecting low molecular weight drugs if you wanted to monitor a therapy. It's actually quite powerful, quite simple, and quite cheap, and has been commercialized by a couple of companies and for specific uses, but the other uses are still wide open opportunities. The second one that we started working on is the fiber optic biosensor. George joined the group in about 92 or 93, right after the Persian Gulf War, and is currently running what's left of the project at NRL. It's been commercialized quite effectively at this point. The way it works is using the evanescent wave. The idea here is that you put your binding molecule on the surface of an optical fiber. The light travels down the fiber and excites only things that are bound to the surface. So if you form a fluorescent complex with antigen and antibody at the surface, it will light up the fiber and the signal will transfer back up the fiber. Nothing further away will generate a signal. So unbound fluorescent molecules don't generate a signal, and there's no signal generated by your sample matrix. Unlike many of the lab-on-a-chip systems, we were interested in detecting fairly rare events with this system. So we didn't want to make any of the holes too small. So they're all millimeters. So you can put sludge over it, or whole blood with no separation, or ground up food, or whatever you want, and it's fine. Just as long as you keep out the sticks and bird feathers, you can analyze it. So it operates within minutes. It detects from 4 to 8 things at a time. The sensitivity is really a function of how good your binding molecule is. So the better, if you're using antibodies, the better the antibody, the more sensitive the system. The other thing that does affect it is the viscosity of the sample. So if you're running a sample like a food homogenate over there, you don't want a 10-minute assay. You might want a 30-minute or an hour assay just to allow the target to reach the surface of the fiber. But the sensitivity is pretty good even with 10-minute assays. This shows a nanogram of staph enterotoxin B, a food poisoning agent, that can be detected with this system. Detects about 1,000 bacteria, anthrax, spores, or whatever. But it has been field hardened, as Paul mentioned, mil-spec. You can drop this thing on all corners. You can take it out in the rain. You can do, well, as I had one Marine tell me once, there ain't an instrument so tough I can't break it. So that's who it was made for. And this, the antibodies are actually, or whatever your binding molecule is put on the fiber. They're put in a cassette like this that has a program on the back that tells the instrument what program to run, what it's looking for, how long to run each sample, and all of that's automated into it. The coupons are stable for almost 2 years in storage at room temperature before they're used. This shows a Marine testing drinking water going on Navy ships in Bahrain. There, the water comes from a desalinization plant in a truck before it's loaded on the ship. So that was considered a point of vulnerability for a terrorist attack. So we had Marines out there putting samples of the drinking water into this device. You can get quantitative information if you hook it to a laptop or telemeter the data to a computer, but they're not interested in numbers. In most cases, this was designed for people who are more worried about lead poisoning than food poisoning. But it actually will read out the answer on this little screen. It says anthrax, yes, ricin, no, botulism toxin, yes, whatever. The first version actually had a little voice on it that told them the answer, so they didn't even have to look at it. And we were teasing. We said all it should say is anthrax, run. But they took the voice off because they didn't want to upset too many people. This Marine was actually quite a creative individual. He got very tired of putting in samples that came up negative all the time. And we couldn't ship positives through the mail, right? So what he did is he went out and ground up some castor beans and shoved those in, and voila, ricin positive. So it is pretty darn robust. We had used the previous versions out in the field even before this system was made. This is George Anderson taking a field trip to lovely demilitarized zone in Korea. In this case, we were using homemade fluidics, and it was hooked to an air sampler, so it would do continuous air sampling, and every 5 or 10 minutes it would shoot an aqueous sample from the air sampler over the device. This shows a similar one that we made up to fly on an unmanned plane at Dugway. Although we're at the Naval Research Lab, I seem to do most of my field testing in the desert. But this was the world's first RAM air sampler. You can see it sticking out the front of this little plane. This little plane's only about this big. So as the plane flies through the air comes in, it spins around in this tube, which we made, using something you get at your local hardware store. All the particles fly to the outside of the tube and drop down in the water, and every 5 minutes it runs through the device to do an assay. And it can do assays over the fibers again and again and again until you fill up the antibodies on the surface. So we actually demonstrated in 86 and 87, again in 87, that you could fly a plane out into a cloud of bacteria, and I want to make very clear this is an anthrax simulant, not anthrax, out in the cloud. Even if you didn't know it was there, because it was all done in the dark where we couldn't see releases happening, and demonstrate that you could collect, identify, and radio the data back on what was there before it reached you. Okay. A report came out about 4 months after the first time we demonstrated this technology that it would be at least 4 years before any kind of remote sensing could be accomplished. Fortunately, we hadn't read the report before we went and did it. That technology has also been used to look at E. coli contamination or sewage contamination of bathing beaches with the EPA. It's been used to detect E. coli 0157 in hamburger meat, and a wide variety of other kinds of applications. It certainly does whole blood, serum, sputum, saliva quite effectively, and you don't have to worry about doing any kind of pre-processing to the sample except possibly adding a little buffer to reduce viscosity or adjust pH. The next technology I'm going to, and Research International is the main manufacturer of that technology now. The next one I'm going to mention is our NRL array biosensor. The fiber optic sensor was only capable of doing 4 to 8 samples at a time, and we wanted to do 40 or 80 different targets at the same time, which is why we developed this technique. It's currently being manufactured or developed for manufacturing by 3 different companies. Hansen Technology is focusing on food applications and some biodefense work. Constellation Technology has focused on biodefense and homeland security. They actually made a version that tested letters going through a letter sorter in the post office and showed that if there was a half a gram of a BG of anthrax simulant in a letter, they could pick it up as the letter went through in under 10 minutes. Unfortunately, they didn't have the lobbyist in Congress to get the award for the monitoring the mail facilities, but they'll go on to do other things. The one you all are most interested in is Precision Photonics. They've developed a very simplified version of the instrument I'm going to show you, and they are using it for AIDS and malaria in third world countries. And they've done over 1,000 actual clinical samples at this point and are very happy with this technology. I've not actually seen the device that they intend to field, but I know that they will be shortly sending it to Brazil and Zimbabwe. The way this works is as follows. You take a very high-tech optical waveguide, also known as a microscope slide, and you attach Avidin to it covalently, and then you immobilize whatever capture you want with a biotin molecule on it. We've done a lot of work with antibodies. We've also looked at antimicrobial peptides. You could use DNA. You could use carbohydrates, which mimic a mammalian cell to get families of infectious disease compounds and so on. So you shine a diode laser. This one's green. We use a red one. At the end of the microscope slide, the first 2 inches of the microscope slide serve as a mode mixer to distribute the light over about a centimeter square area at the far end of the slide. That's where we immobilize our capture molecules, and we then take an image of the illuminated spots at 90 degrees, and the location of the spot tells us what was there. We have multiple ways of making these spots. This is the poor man's way, or one you can do in any laboratory, where we simply make a block of channels, and we put our biotinylated capture molecules in this direction, and then we flow the samples in this direction. So you can do multiple samples simultaneously for multiple things. You can include positive controls in every sample, which is what you see here. We included a fluorescent chicken IgY in every sample and have an anti-chicken IgY at places we can use for registration marks and to show that the registration marks for our automated program that analyzes the spots, and just to make sure the system is working. You also have areas between each spot where we take data, and each of those areas has seen the sample, it's seen the fluorescent reagent, so you have a check for any background fluorescence that you might get, and also some control for any discontinuities of fluorescence across the slide. This is what the NRL version looks like. It has a commercial camera here. This is the diode laser. This is the main cost of the system that we made because we're taking off-the-shelf components, but we've also showed that you can use a CMOS chip to do the same thing. Or if you wanted to, instead of buying a $7,000 camera, you just take a chip and work from scratch with your own chip, cut the cost significantly. We have put all the fluidics part on this side of the device. There is a partition between the electronics and the fluidics. This is very important. We learned the hard way that water and electronics don't get along too well. So we have pumps and valves over here. We have reservoirs that could be plastic molded. We're still putting together by milling. but one holds 6 samples in this case, and the other holds 6 sets of fluorescent reagents, which we've demonstrated can be freeze-dried, stored, and added later. But this is still a fairly big version of what is possible with this technology. Yet it's still light, portable, works off a battery, and so on. Like the fiber optic system, it uses an evanescent wave for illumination and therefore is very immune to interference from fluorescent molecules in the sample. We've demonstrated that it works for a wide variety of clinical fluids. This shows clinical fluids spiked with the F1 marker for plague, normalized to what you would get in buffer. The signals are normalized here, but what it shows is that even at the very lowest relevant physiological concentration, you can detect it in all of these samples with virtually no sample prep. The intensity of the sample the signal varies a little. We love nasal swabs, nasal wash. We get what we call the SEE, the snot enhancement effect. For some reason, very little nasal material seems to concentrate our target very nicely and gives us a beautiful response. We've never been able to figure out exactly what component in snot is doing that, but it's very reproducible. We've also looked at a wide variety of environmental potential interference. We can spike in a % of sand or clay or hydrocarbons from smoke or dyes or whatever, and it just doesn't make any difference. So it's very robust. These are some of the limits of detection we've gotten with antibody assays. And the only 2 points I really want to make here is that the variety of things we can detect, including both low molecular weight toxins and high molecular weight toxins, both gram-negative and gram-positive bacteria. We've done a few viruses, but we're mostly BL2 facilities, so we're limited. But the sensitivity is really a function of how good your antibody is. So the better the antibody, the higher the sensitivity. With our best antibodies, we can get down into the picogram per mil level of these materials. And generally, we're testing about 400 microliters. So those are the ones that are becoming commercially available. The last one I want to talk about, I want to particularly give John McDevitt a thank you for introducing the concept of flow cytometry. Unlike the way he's doing it, we're trying to actually miniaturize the flow cytometer, which is, I know that Paul has also done some work on as well. But what we've done is we've gotten most flow cytometers on a chip take buffer streams to focus the channel under the light. Okay, so they take one from each side, and they basically, what you have is a rectangular section in the center. To get that focused in both the X dimension and the Z dimension becomes much more complicated because it's very hard. You either have to use dielectrophoresis or additional inputs of fluids to do that. What we've done is we've come up with a way that's patent pending, Paul, patent pending, where we can take 2 streams coming in, one from each side, and entirely wrap one stream around the other using passive mixing devices in the walls of the channel. So this actually came out of microfluidic mixer studies that we've done that allowed us to control the systems. The other thing you can do is by changing the ratio of the flow rates in those 2 fluids as they come in, you can focus the channel down smaller and smaller. So this just shows what one of these devices look like. And it's very important that you're not getting a narrowing. These channels can be fairly large if you want, so that you can put fairly complex samples through this. We've made these everywhere from 100 microns in diameter to a millimeter in diameter, and it still works quite fine. You then wrap one stream around the other, and what you get is a focusing of the central stream down the middle of the channel. Even something like a bubble or a piece of dirt caught in the system doesn't prevent you from being able to do this. So again, it's pretty robust. This just shows that we can actually focus the channels down up to 42,000 times in terms of the volume to volume ratio. So we can get that core very, very small. Again, so we have all our particles, blood cells, whatever you want to detect, lined up in a nice row. The only limitation on that is the pumping mechanisms that you have to use to pump in the 2 fluids. We've mostly done our work to date on beads. We haven't actually done real assays yet. Those are scheduled for next month. But we've shown that we can distinguish fluorescent and non-fluorescent beads, and that the count of fluorescent beads, even at very low numbers, reflects what's actually being put through the device. The other thing that's really nice about this system is you can reverse those structures in the wall of the channel. So you can separate the core stream from the sheathing fluid. This means that you can have a self-contained system. You don't have to keep adding lots and lots of sterile water, and allows you to put it in places you might not otherwise, and to run it unattended for long periods of time if you want to do continuous monitoring. I mean, that's not the format you would use necessarily for clinical monitoring, but if you want to monitor the environment for cholera in the drinking water or something like that, you can do this now without a large logistical burden. As I was mentioning earlier, I'd like to put this up on the space shuttle to test recycled drinking water. If you're going to drink water that is recycled, you sure would like to know what's in it or what's not in it. We can also use it for continuous monitoring with air samplers to detect bacteria or viruses in air. The flow cytometer, the Luminex flow cytometer, has actually been used for that in subways, including DC, and the system is about the size of a podium like this, and a third of that is water. So this allows you to recycle the water and make a very small system. So it does have some very unique features. We can also build it with reagents on chip for multiple assays. This recycling means that if you wanted to capture the target that you identified for genetic analysis or antibody resistance analysis or some other thing, you can capture on chip the target that you were interested in. So I'm going to conclude just by, this has been kind of a survey of some optical biosensors. Some of them are available for point of use. In the near term, the focus has been on expanding the sensing capability, the number of things you can detect at the same time, and miniaturization and automation. Miniaturization is certainly critical for automation. And the future is focused really, again, on the inexpensive, easy-to-use sensors. Remember, the people who use the sensors that are my biggest customers may often have a high school education. So that's the top end of what we're looking for. But at best, they're too busy to think about what they're doing. We're also looking into biosensors for distributed operations, multiple targets, continuous monitoring, and networked response. Thinking about these as systems takes on a whole new dimension if you think about making them cheap and small enough and networking them together. That technology is available if we can bring the cost of the sensors down. And it becomes very important for epidemiological surveillance as well as other kinds of surveillance. So thank you very much for your attention, and stop here.
Maybe if we can get the panelists up here, and we'll maybe answer a couple questions before break. Yes.
Let me congratulate the speaker for their brilliant presentations. My question is to John. You've done well with the lab on a chip for CD4 estimation, and I'm sure that will make things easier for us in terms of starting treatment. But there's still a critical missing factor. When we start treatment, we need to measure progression of the viral load, which is the PCR RNA. Do you have anything in the pipeline to measure that?
That's a very good question, and we do have two efforts ongoing to look at viral load. We chose CD4 first for a couple reasons. A practical reason, it's easier for us to do, number one. And number two, in the homework that we did in interfacing with people in Africa, the feedback that we got was if you could only do one first, then to do the CD4 first. For viral load, we have a couple things that we're looking at. One is actually analogous to what Paul had talked about. So on-chip PCR measurements that would put this basically the same way that it's done in developed countries now, but miniaturize that onto a chip. That has some additional development challenges that will take 6 months to work through. We have a faster alternative that we're exploring now based on a cellular surrogate test that has some correlation, some decent correlations with viral load, but it's not FDA approved, and so there would be a balance relative to how quickly that would be available, and it's probably not an approach that would be viewed as something that would work well in the States because of the regulatory challenges. But given a lack of any information, this seems to be an acceptable alternative that we can have ready relatively quickly.
I have a question for Rebecca. What's known about the safety and persistence of the nanoparticles in tissues?
So it's different, of course, for every kind of particle. And a lot of the emissive particles that we're talking about very well, most of the applications we're looking at ultimately would be in vitro, not in vivo. I'm really not convinced that we'll be able to use any of those in vivo. But the gold-based ones, I think, will be able to be used in vivo, fairly tolerant, really good clearance. We were talking about really small volumes relative to what's been used before. The dose that's being talked about, it's a 50 you have to scale up from a mouse, but you're talking about it 50 microliters at about 10 to the 9th per mil. So it's a very small amount, and it's just a gold particle. The reason you use gold rather than some of the other things like silver, which have much better optical properties, is basically because of the biocompatibility and how easy it is to modify the surfaces.
Any more questions? Maybe.
How do you communicate with one another? I mean, many of you are involved in collaborations, and then there's a conference like this one. I don't know how many such meetings there really are, and it's kind of hard to keep track of all the literature. Is there a well-defined network of researchers and users across that whole spectrum that makes it fairly straightforward to keep track of what one another's doing? And
I think it's kind of ad hoc. It's who you know, who you meet at conferences, who you happen to put proposals together with. Certainly there's a lot of internet traffic with people you're actually working with, less so with people you're not, but visiting. I come down and visit John once every couple of years. You haven't come to see me yet.
You haven't invited me.
We'll fix that.
In other words, how do you avoid overlapping efforts and duplication of your energy?
We don't. But the thing is, no two people will. If you have a single problem, even with a single technology, no two groups are going to do it the same way. And so each one's going to have advantages and disadvantages. And there's no one single answer to this problem. It's going to take a lot of different answers to be I mean, it's such a complex problem. So there's plenty of room.
And let me just add a little bit to what Fran says. I think there is some thought behind the scenes to create complementary efforts. And for example, the NIH has a cooperative research program, UO1, that Paul and I were involved in. And looking at what happened in that particular program, there was not a direct communication, but just an evolution which occurred so that each of the groups that had the same goal of miniaturization, using lab-on-a-chip systems for saliva-based diagnostics, but by virtue of different strengths and weaknesses for the various approaches, the targets remained different, complementary and synergistic.
So on that note, maybe we'll just go to break and just continue the discussion.